whenever you first walked in here, you should have been handed 2 scratch and sniff stickers, one objectively good and one objectively bad. You can smell them now. In life, there are some known bad smells, such as wet dog, and also the smell of sewage treatment facilities. There's also some good smells, like the smell of roses and the smell of freshly baked goods. But there are also some smells where it just depends on who you ask, like the smell of gasoline and the smell of Sharpies. This might be the same case for your stickers. You might think something smells good, but your friend might think it smells bad. Over the summer, I did research at LSU in a lab to test the same question, but instead of using humans, I used flower beetles. Flower beetles, also known as rice weevils, are a type of infectious insect that they infiltrate inside of flour mills, and they eat the flour and they lay eggs in it. This makes the flour inedible for human consumption and also unsellable for the flour mill companies. Obviously, this is bad because whenever you eat, you don't want to have crunchy beetles in it, and companies can't sell anything that's infested. There are 2 types of flower beetles, confused flower beetles and red flower beetles. You might not notice that much of a difference between the 2, besides that confused flower beetles have slightly bigger proportions. But I wanted to test to see if they had any other differences, like their preferences for smell. The smell I wanted to test was the scent of Beauveria bassiana fungi, which is a parasitic fungi commonly used in integrated pest management practices because it has high mortality rate for insects but has no harmful effects towards humans. This is what it looks like whenever an insect is infected with the fungi. The fungi gets inside the beetle and kills it on the inside out and sprouts spores on the outside of its body to infect other insects that come in contact with it. In my experiment, I used an olfactometer, which is practically a scent machine. It pushes air throughout the tubes into the jars and into the base of the experiment, and base of the olfactometer, making the scent of whatever in the jars push inside the base where my beetle will be. Inside the jars is 10 grams of flour, 100 beetles, either red or confused, depending on which one I was testing at the time, and 1 gram of the fungi, making that my experimental group and my other one the control group. Inside of the machine, the beetle will be placed on the center on the X and then timed to see how long it takes to choose one of the sides, either control or experimental group. My hope is that the beetles will be repelled by the scent of the fungi and go towards the control group. The results of this experiment were that for the red flower beetles, for both male and female, they had around a 50% chance of choosing either or. But for the confused flower beetles, they had a higher than 50% chance of going towards the control side. Meaning, given the chance, the confused flower beetle has a higher chance of being repelled by the scent than the red flower beetles. On this map, you can see where all the dark spaces are, that's where the flower beetles are infested in the world. They're almost everywhere on every continent, and they also are very resistant towards normal pesticides or other methods of getting rid of them, which makes it important that we find a method that is both effective but also can keep the flour that we use safe. In my experiment, I wanted to find a method that could possibly repel the insects from the flour before it even gets there, so that we don't waste flour products. Because after the insects get inside the flour and you have to use pesticides to kill them, the flour is unusable. I would like to give thanks to my Dr. Cronin and his PhD student, Aaron, who helped me throughout my experiment and let me use their lab, and also like to thank Dr. McLean, who helped me give this opportunity to do this research through eStar. I hope that in the future that we can find a method that is both effective for both red and confused flower beetles, and so that a method that's effective for both confused and red flower beetles, and that we can keep our flowers safe. Thank you.